Assalamu alaikum, myself Talha Salim, and the topic which I am going to discuss today is heat shock proteins. As you all are aware of the proteins, so let me just tell you a bit information or like a short description about the proteins. So the proteins are the building blocks of human body, and proteins are made up of amino acids. The amino acids are joined together by the help of peptides. Therefore, the chain of amino acids are also called as polypeptide chain. The proteins are of different types, such as primary, secondary, tertiary, and quaternary structure. In case of primary structure, the proteins are linear, as you can see here, and the secondary structure are basically the helical, like in case of DNA, and the secondary structure are either alpha helix or beta plated sheets. The tertiary structure is like a fold, 3D fold of secondary structure, and the quaternary structure is like when two tertiary structures combine, they form the quaternary structure. This quaternary structure could be homoquaternary or heteroquaternary, based on that if two same quaternary structures are combined, then they form homoquaternary structures. And in case of heteroquaternary structures, the tertiary structure which are joined are two different basically. So let's not go into further details of the protein structures, and now let me tell you about the heat shock proteins. The heat shock proteins, also called as stress proteins, because they are usually developed or they are produced in response to stressful conditions. These stressful conditions could be like cold, UV radiations, healing, like if somebody is injured or due to an injury, the heat shock proteins are produced in the body. And these heat shock proteins, like they help the body to prevent the protein misfolding, or like if certain proteins are misfolded, then these heat shock proteins help the body to like degrade the misfolded proteins by taking them to the recycle bin, dustbin, such as proteasomes. And after that, heat shock proteins like discovery, which is the second slide, is the discovery. So heat shock proteins were first described in the early 1960 in Drosophila by Ritusa. Basically, they were first discovered in Drosophila, so that's why. And the heat shock proteins are classified usually basis on the basis of their molecular weight, such as heat shock proteins 60, 70, 90, and 100. Also, there are certain small 8 kilodalton proteins which are called ubiquitin. These are like the markers. Basically, if certain protein is misfolded, then ubiquitin are basically attached there. So basically, they are giving the signal to the other heat shock proteins that this protein should be degraded. And also there are certain other heat shock protein types, such as the small heat shock proteins, which are very small, like 80 amino acids, alpha crystalline in structure, and they help in assisting the other heat shock proteins for the degradation of proteins. The functions of heat shock proteins are different, and one function is like stress response, in which, like, if your body is facing certain stress conditions like an infection or inflammation, then these heat shock proteins are there, because they will then help your proteins which are present in the body, and they basically attach to the proteins, so the proteins should not be degraded, or that the proteins are not degraded after the stress conditions. So they help, or they basically prevent the protein degradation in stress response. The other role is the role as chaperone. Basically, the small heat shock proteins also act like chaperones. Chaperones are like assisting the degradation, or like they also confirm the shape, like if certain protein is properly folded or not. So that's basically the function of chaperones. So like as you can see in this picture, the proteins are basically attached to the receptors, and then as they enter in the mitochondria, the chaperones are basically these green ones, and the blue ones are the chaperones. So basically they starts removing, and after the conformation they are again attached. So their attachment and removal requires ATP. And the next one is the cellular stress response. The cellular stress response is that, as I told you, that in case of certain proteins which are degraded or degraded, with certain proteins which are misfolded, then these if stress proteins, which are heat shock proteins, carry these misfolded proteins to the recycle bin, and this recycle bin is proteasome, where the proteins, misfolded proteins, are degraded. So that's why these proteasome are called recycle bin of proteins. In case of any infection or injury, the infected cell releases certain proteins which are not normally found in the body. These proteins are the signaling proteins, and they act as an antigen, due to which the immune response or immune system is provoked. After that, the immune cells starts the im- attacking the cells, and also heat shock proteins are produced, and they basically alarm the whole immune system. So the immune cells are also triggered by the help of heat shock proteins. Lens, as in case of zebrafish, so the alpha crystalline or HSPB4, sorry. So the HSPB4 proteins are involved in the development of lens in zebrafish, in its developmental stages. And now the question arises that why do the heat shock proteins are not denatured? So there are four reasons: that they have better hydrogen bonding, better hydrophobic internal packing, enhanced secondary structure, helix dipole stabilization. So these four are the reasons why heat shock proteins are not denatured under a stressful condition, because heat shock proteins are also proteins, and proteins are degraded at high temperature or certain temperature. So these are the reasons that heat shock proteins are not denatured. These are the references, and thank you. If you have any questions, so please do ask.